Hi, I see some known faces here. Good. Well, thank you for joining. Good morning for those in other continents. Good afternoon, and welcome to this IHI's Realize the webinar entitled Evidence Assessment Framework, the need for mindset shift among developers, regulators, and HTAs. Before we move into today's discussion, I would like, if you don't mind, to pause for a second and express our deepest condolences to the victims, the families, and all those affected by this tragic train accident that occurred in the south of Spain less than 2 days ago. My name is Alicia Granados. I'll be chairing today's session jointly with Alex Korskaden. Sorry for my pronunciation, Alex, that will support us, but also will manage the Q&A section. We are pleased to be joined by an outstanding panel of speakers, Francois Meyer, Andrew Thomson, Karen Facey, and Florian, who will bring deep experience from regulatory, HTA, methodology, and patient perspectives. And together we will explore why a shift in mindset is required and how evidence is generated, assessed, and used across the ecosystem. Next slide, please. Before going or briefly introduce the RealiseD project, I would like to share with you the agenda. We will have some housekeeping rules. I will make a short introduction about the RealiseD project. Be aware that this session will be recorded, and by participating, you consent to the recording and any use for educational and promotional purposes. Next, please. And then the typical housekeeping rules: mute your microphone, use the Q&A function, but feel free to submit any questions and comments via Q&A function. And of course, when it's time for the Q&A, raise your hand. Next, please. As I mentioned, I'm Alicia Granados, and I'm serving as Global Medical Affairs in Rare Disease Sanofi, and also RealiseD work package it's co-lead, jointly with Kit Rose from Radboud University, that today is not able to be with us, but is in our heart. Well, next, please. Yeah. Well, as I mentioned before, I introduced the RealiseD project, we will like to learn a little bit more about you and your perspectives. So please connect this Q&A code. It is a very short and anonymous questionnaire that is designed to help us to understand who is in the audience today, and more importantly, what your main concerns are when it comes to evidence in rare diseases. Your responses, Alex, you can share, will help to inform today's discussions and future work within RealiseD. And also, if you use this, so we can see live who you are. Interesting. The interest of industry, HTAs, patient advocates, academia. It is, do we know, Alex, what is the percentage of the answers? Okay, 24. Okay, we see, we see here in the slide. Should we go or should we wait? Alex, you are
We can go ahead to the next question.
Okay. Next. What do you think of evidence in rare disease? What is the first word that comes to mind? Let's vote. It's an open question here, no? No. Not a lot of participation. I really encourage you to participate because it will be helpful. 6. Okay. Let's go next. Which design do you consider most credible in ultra-rare diseases? Interesting to see. You think we can go next to give? I think people can keep voting, Alex, or not?
Afraid not. Once I move on to the next question, the voting closes.
Okay. But it's been a good experiment anyway. Next. Where do you see the biggest divergence between regulators and HTA bodies in rare diseases? Acceptance of uncertainty, endpoint selection, use of real-world evidence, magnitude of effect required, consideration of patient-reported outcomes, meaningful expert input. Okay. Move. How confident are you in real-world evidence to reduce uncertainty in rare diseases? Very confident, moderate confident, low confidence, only in a specific context. We have a small n, taking into account that we learned that we had 200 registers, something like this, but well, it is something. Next, how many more are?
There's 2 more.
Okay. Should IGOR uncertainty be acceptable for therapies targeting very small population? Yes. No. Okay, only when there is transparency over the scientific, biological, or ethical rationale for not providing randomized. No. Okay. So next. Two current frame. Okay. The strive for right balance between rigor and access. No. Looks like unsure. People is unsure. So. And I think the last one.
That is the final question.
This is the final question. So we have a roughly idea of what is our audience here and what are their main concerns, even though we prepared this webinar without knowing this answer. So let me, next please, let me set briefly the scene by introducing the RealiseD project, which is a scientific consortium. It's a public-private partnership initiative with around 50 EU partners that has been built to respond to some of the known challenges patients are suffering from rare diseases are facing for accessing optimal treatments. We have an ambition that is maybe this group of scientific organizations is going to develop cutting-edge operational and methodological tools and resources to advance both evidence generation and the evaluation of new treatments. Next. This is just an image of how this 5 years project that started in January 2025, so we have a year running, and you will see that there is academia, European reference networks, several companies, HTA and regulators, including EMA and EQUIC and Critical Path Institute, and other research organizations and patients organizations, of course. That's just to give you an idea of the dimension of this consortium. Next. The structure of, and I don't want to enter into detail because in the first webinar it was extensive explained, but the first thing is we want to align with the ecosystem. This is an initiative and we know that we are not the only one with these concerns. So we are first mapping who else is doing things in this regard. So, and trying to align with several initiatives already described in the previous webinar. Then we have the ambition also to develop a more efficient patient enrollment system using use cases, clinical use cases, and how to certify clinical sites. Also, we are dealing with innovative clinical trial designs and innovative analysis of data strategies as well, no? Then we will revisit what are the regulatory and HTA perspectives, linking this with development, we can do together something better in order to improve patient access. And of course, patient is engaged in all the work packages that are 11. And of course, then we like to develop some innovation readiness playbooks in order to provide resources for those who want to use all the work that we are doing. Next. And the pillars. The RealiseD has at least six strategic pillars that collectively drive innovation and impact. Again, it is about improving clinical trial design and execution, about multi-stakeholder collaboration, I would say beyond collaboration, co-creation, statistical innovation, patient-centricity, regulatory HTA and industry developers dialogue, and clinical use cases. Next. Let's go to the objectives of the webinar. We have been spending maybe too much in the introductory part, but the objective of the webinar is to dive into the current European regulatory and HTA landscape, highlighting what's going on in Europe and beyond, and to overcoming the challenges we identify, we know, but also that our audience were putting in the survey. We want to also learning from these experienced experts in methodology, regulatory, and HTA science, and hear the perspective, of course, of the patient advocate, which is our main objective of all this work that we are doing. And finally, trying to showcase how this consortium could advance in scientific solutions while we all together advocating for a fundamental transformation of the way evidence for rare disease is generated, interpreted, and assessed. Okay. Here are our speakers. I said that this RealiseD project is rooted in a simple but critical observation. Today's evidence framework used by all of us are often insufficiently aligned, especially in ultra-rare diseases, and that this misalignment generates uncertainty, delays, and ultimately unequal patient access. To describe the current situation in Europe and explore challenges, it is my pleasure to introduce Francois Meyer. Francois brings an extensive senior experience from HAS, ANSM, EMA, and has been deeply involved in shaping both national and international regulatory and HTA cooperation. Francois, the floor is yours. Next slide for Francois. Yeah. Can you hear, Francois?
Alicia, I think you may be on mute, but I suggest Francois is not available. Maybe we could move to the next presenter while we wait.
Okay, let's go to the next presenter. It's a pity because we have here the slides from Francois. Next speaker was planned to be Andrew Thomson. Andrew Thomson intended to walk us through very concrete design choices developers and others face, and particularly in ultra-rare diseases, and how different evidentiary strategies may be perceived across a stakeholder. Andrew, over to you.
Thanks, Alicia.
Thank you for the flexibility.
Next slide, please. So I wanted to start by thinking about now what happens in kind of standard common disease adult de novo development. And the way the trials are usually planned is that regulators set this hurdle on the left-hand side of P less than 0.05, and that's what's expected that developers are going to clear. And then developers have a choice. They're weighing up the options in front of them. Usually that's for a given effect size that they're interested in targeting, picking a particular sample size in order to guarantee the power that they desire for their study. Now, that's an excellent way of doing things in common diseases, but it also assumes that the patients are there. Next slide, please. And underpinning this idea and the key assumption is that the patients are there, and also that the patients want to be in this trial that is being proposed. And they're not necessarily the same thing. Sometimes in ultra-rare diseases, there are just not enough patients globally, whatever the trial design, for the usual package of evidence. But it's also got to be attractive for patients to take part in as well. And also, we must reflect as well that the industry have to want to do the trials. They would like to make a return on their investment, which will depend on how much they think they're going to sell it for, how many patients are going to be eligible for it, but also how much the trial costs to run as well, how much the investment's going to have to be. So it's not just sales, it's the cost of investment as well. Now, when we move away from that, and say, well, actually, these trials are not going to be possible, the patients aren't out there, then now we have to start making some different decisions. And one of the slides in the multiple choice earlier was about the kind of like different strengths of evidence. And I'll talk about this as an ex-regulator on how regulators might think about interpreting. Next slide, please. So there's four big ones that I can talk about today. Some of the others, such as adaptive and Bayesian, I probably haven't got time to go into. I wanted to focus briefly on 4 possible options and the pros and cons from a regulatory perspective. Obviously, other people would have different pros and cons for them as well. And they are alternative endpoints, running a clinical trial bigger than the usual value of 0.05, external data incorporation, which can be Bayesian or it can be frequentist, but oftentimes it's thought about in a Bayesian way, or indeed single-arm trials. Next slide, please. So one possibility is to run the clinical trial using not the clinically relevant endpoint, but to use something, say, for example, a biomarker or an alternative endpoint, and then demonstrate some sort of trend or meaningful effect size, even if it's not statistically significant, on a clinically relevant endpoint as well. Now, the pros of this include that you get this evidence at p less than 0.05, and so everyone's clear that the drug is doing something. It might not be the clinically meaningful endpoint, but it's certainly having an effect on the body, and hopefully there's a kind of trend as well to go with it on a clinically meaningful one. This is simple, it's easy to understand, and I've put easy to agree with the regulators, and this is in quote marks here because obviously all things are challenging to agree with the regulators, but what I mean is the amount of justification that goes into this is not necessarily quite as much as some of the statistical methodology justifications required for the more complex statistical designs. Potential cons are trial burden, whether patients want to go into the study if too many things have been measured. How you define this trend on the clinically meaningful endpoint, does this need to be agreed with regulators, what the evidence standard is every single time as well? But I think also this endpoint needs to exist. Everyone says, oh, yes, great, let's use biomarkers. And then that assumes that there's one out there that you can actually use. And that's not always the case. And finally, I think validation is important, and I don't think validation is going to be easy here by the usual regulatory standards, because there aren't enough patients to do that in advance of the trial. So to some extent, this kind of alternative endpoint will have to be validated through the trial itself as well. Maybe that's the price you have to pay for the rare disease. Next slide, please. You could just use a higher p-value than 0.05, and again, that's simple and easy to understand, and potentially the justification for it is straightforward. I'd like to point out this is in line with the EMA small population guidance, and also ICHE 9, which does say that different values of p compared to 0.05 can be used. I'm not sure if the original intention was that they would be smaller, but certainly the door is open in both of those documents for this approach. Nevertheless, it might not be acceptable to downstream decision makers, and I don't just mean HTA, I mean prescribing physicians as well. Sometimes it may be difficult to get a paper published if reviewer 2 comes back and says, well, I don't care if you agreed a p-value of 0.12 with the regulators, I think this is a failed trial. And then it's difficult to get it published. People like prescribers might not find the evidence convincing. I think it's also challenging perhaps to get global harmonization. Maybe FDA would say, well, we'll accept a trial at 0.08, and EMA say, well, we'll accept one at 0.12, or vice versa. And you kind of automatically have to default to the 0.08, the lowest common denominator, just to make sure everybody's happy. Next slide, please. Maybe you could borrow external data, and I'm thinking here about a hybrid design. So there's still a randomized element, but maybe there's some external data, oftentimes to kind of increase the control arm. One of the benefits here is that you can obtain a level, maybe not a p-value, but some evidence level of 0.05 on a meaningful endpoint, and that can be convincing to others, downstream decision makers as well. I hear from some statisticians that Bayesian methods are simple and intuitive for people to interpret. And so I've put this easy to interpret with a question mark. It's possibly also difficult to interpret as well. These models are oftentimes very complex. There's a lot more assumptions out there that may or may not be true, and there may not be the data to justify them. Ultimately, anything that borrows external data assumes it exists. And again, that might not be the case, especially in ultra-rare diseases. These data sources might not actually exist where you could reasonably borrow some information, but maybe real-world evidence in well-conducted registry studies could provide some useful information. Next slide, please. And finally, single-arm trials. They are a possibility, and they are definitely simple, and they are easy to understand, although sometimes challenging to interpret. I would say that these are potentially in line with EMA guidance. EMA has a reflection paper on single-arm trials, and they explain when and how they can be used and what needs to be done to justify their use. I think another benefit of single-arm trials is that they can be contextualized with external data. So you don't necessarily need to make a formal statistical comparison if you're not entirely convinced that the external data is completely fit for purpose to make a statistical comparison. It still exists nevertheless, and you can think about the trial in the context of the existing knowledge. And finally, another pro may be the patient preference. We sometimes hear that patients prefer to be in studies where they're not going to be randomized to placebo or not going to be randomized to what they might consider to be substandard care, which of course is entirely reasonable. And so single-arm trials may be more desirable for patients to take part in, but maybe that's a case-by-case basis. Again, that might not be acceptable to downstream decision makers, and maybe HTA are quite important here, especially if there's no comparative evidence to anything, then that might not be acceptable. And again, global harmonization may be challenging between different regions. And actually, if it's a single-arm trial, it's not easy to rescue it. It's such a fundamental difference in the design. We're not arguing about p-values or which Bayesian parameter you want to tune, but actually whether there exists any randomized controlled data or not. So if it's not acceptable, then it's hard to rescue. Next slide, please. I think all of these sit on a scale of strength of evidence. I think, and this is through a regulatory lens that I apply as an ex-regulator, and I would say that the kind of the biomarker plus alternative variable is more convincing than p less than 0.05, simply because you have more information. You will have that clinically meaningful endpoint as well. And that and the hybrid trial, I think, are similar but different. And the SAT is less convincing on its own. And the reason I've put the p-value and the hybrid trial in the middle together, because ultimately what I see these as is two sides of the same coin. And actually what needs to happen here is a trade-off between having a unbiased but underpowered with a large variance trial, or having something that might be more powered with a smaller variance but could be biased. And this in statistics is classically known as the bias variance trade-off. So whether which one of these two you would like to do might depend on your views on bias and variance. Next slide, please. So in the spirit of the webinar, I just wanted to finish with what is the challenge for regulators. Well, we've moved actually from the pharmaceutical industry deciding whether it's sample size and power that they're trading off to actually regulators having to decide whether they would like to trade off bias or variance if they want to study at all, because if they don't make that trade-off, then patients and industry may not be providing them with the data they want. And so what regulators can do is explain their preferences, like the single-arm trial guidance. What do they want? When do they want it? What are the situations when each of these different biases and variants can be traded off, and why that's the case as well? And I think if we do that as a community, then we can actually have better guidance on what trials in the future need to look like. So this is something that can be done now to help define the needs of trials of the future. Thank you.
Thank you so much, Andrew, for these examples that has illustrated how differential trial designs and evidentiary approaches can shape regulatory decision-making, but also downstream challenges. Of course, this naturally raises the question of how such evidence is interpreted, valued, and assessed once it enters in HTA process. The problem here is that the plan for the webinar was starting with Francois, describing, I see him here, describing the context of current regulatory and HTA requirements, then going to Andrew with methodological and regulatory mindset, and then going to Karen, and then going to the patients. But let's be flexible. And I would like to ask Karen to go into the HTA because the flow is going well. And before going to the patient, maybe we will do the opposite. And then Francois, before the patient, will give us your contextual approach. I hope you all agree. Karen.
That's fine. Thank you, Alicia. Thank you, Alicia, for giving me this opportunity, and you've made me think by putting together these slides. So if we go on to the next slide, I'm now talking about the next step after the regulators have done their work about working out whether a drug has quality, safety, and efficacy, to think about health technology assessment, which is undertaken by payers and healthcare systems to understand whether a drug should be reimbursed or covered in their own system. And they use this process often called health technology assessment, HTA, and it's used internationally. And 5 years ago, a definition of HTA was agreed by all international HTA bodies and associations as a multidisciplinary process that uses explicit methods to determine the value of using a health technology at different points in its life cycle. Its purpose is to inform policy making and decision making to promote an equitable, efficient, and high quality health system. So should we be investing in a new medicine that comes forward, recognizing that there will be an opportunity cost? We won't be able to spend that money on other patients. And there are two key things about the definition. First, we will look at what it means by value, but it also indicates that the process is formal, systematic, transparent, and should use state-of-the-art methods to consider the best available evidence. And I want to focus on that in my presentation. So what do we mean by value, and what do we mean by best available evidence? And how is that systematically reviewed in this context of rare diseases? Moving on to the next slide. The definition paper itself actually expanded what it meant by value, saying it should be assessing the intended and unintended consequences of using a health technology like a medicine compared to existing alternatives. And this is where we're different from the regulators. We always think about how does it compare to what we already have in our healthcare system, and should we invest in that? But the way every country or jurisdiction undertakes this health technology assessment and pricing and reimbursement process is different. They're all common from the point of view they all look at clinical effectiveness and safety. So will the drug work in our setting? Is it providing added benefit? But then across different jurisdictions, some do costs and economic evaluations. Increasingly, more agencies are looking at environmental aspects. Some look at organizational issues, particularly for more complex products like gene therapies, for example, in rare diseases. Some look at ethical, social, cultural, and legal issues, and others look at the wider implications for patients, carers, and family. And they recognize that actually your overall determination of value will depend on the perspective you take. And I'm interested that there are these dimensions of value. I think they're all very important. And I think that what's happened in recent years is that since manufacturers have been asked to provide the evidence to HTA bodies, systems have focused just on the first element in particular, and maybe the second element if that system requires economic evaluation. And evidence hasn't been provided for some of those other areas. And it used to be the case that that evidence drove the decision making. And I want to explain why I think all these domains of value should be considered with the best available evidence. So if we go on to the next slide, we can see here, let's start with clinical effectiveness. And I learned my HTA from Denmark. And this was a model that was presented in Denmark early in the 1980s by the wonderful David Banter. And we recognized here that we had clinical effectiveness, cost effectiveness, and in the Danish model, there were two other dimensions of organizational issues and patient aspects. And all of these aspects required systematic approaches to gathering the evidence. So clinical effectiveness based on evidence-based medicine using systematic reviews of international clinical research. But then all the other elements were local, gathering the best available information about how workforce systems were set up in terms of patient aspects, doing systematic reviews of what patient issues were, and doing local primary research about the issues that patients face, and then economic evaluation. I'm sure many of you on the call know about that, that's done at a local level. If we go on to the next slide, what we need to recognize is that even if we have evidence coming in from all these sources, that evidence needs to take account of the fact that we have a rare disease population. And what does that mean? What should we be providing leniency in the way we think about evidence because a disease is rare? Well, I would say yes. And this work comes from the HGAI Rare Disease Interest Group, which I'm proud to be a chair of. And this work comes from one of our project groups, where they've identified that rare diseases are complex, but you can't just put all of them in one bucket. You have to characterize what are the specific issues with each rare diseases. So you need to think about the prevalence, because actually you can have quite high prevalence rare diseases, and most of the challenges arise with the ultra rare diseases, and we don't even have a common definition for ultra rare diseases. Also, the age of onset is important. The issues surrounding doing trials in children are well studied in other fields, and we need to recognize those in the rare disease context. And then the things we all know about, that many of which are written in the small population guideline that Emma has talked about, in terms of the challenges of dealing with small populations. But in rare diseases, we have heterogeneous conditions, so we don't know what's the best endpoint to study. They're severe, so it might be difficult to measure endpoints. They're complex, so we may not have a clear disease progression, natural history profile. And often we have scarce knowledge about the disease. I'm sitting here in Scotland, and for many rare diseases, we don't have a specialist. We have to go to other countries for specialists. And we shouldn't call what we don't know about that condition uncertainty, because it's not uncertainty, it's scarce knowledge. And we need to stop calling everything uncertainty and characterize it clearly to understand whether we can, what we need to do to make the evidence better. Moving on to the next slide. I have been really struck by this article, which was published a couple of years ago by Subia in Nature Medicine, and it's really sort of changing the paradigm. If you look at the top of this iceberg, you can see the traditional hierarchy of evidence, which has been the foundation of evidence-based medicine. But look at the bottom of that iceberg, and it shows you where we are in today's clinical world. We have information and innovative science that is giving us all sorts of evidence that we could be using in our evaluations of whether a product is working or not. And we need to be bringing all of these into consideration, particularly for rare diseases where we may have smaller clinical studies. Moving on to the next slide. Another paper that has been really striking to me, published at the end of last year, was by Keith Abrahams and a number of colleagues, including Hans-Georg Eichler. And he looked particularly at cell therapies and gene therapies and particular case studies to identify that question that Andrew alluded to about when are single-arm trials feasible. And you'll see in the center of this slide here, the development program factors. And there are some situations where actually RCTs are more feasible, and there are some situations where they're less feasible. And if you look on the left-hand side, you'll see where RCTs are less feasible. And that's when there are small populations, rare diseases, patients difficult to identify, fatal or progressive diseases, large effect size, long time to effect, no effective standard care, complex and burdensome treatment. And what I'd like to be doing is taking some of those domains and putting them alongside the way we characterize in the RDIG a rare disease. And we need to be kind of explaining these issues of characterization of rare diseases that mean we can or cannot do a specific trial design. And that should all be critically assessed. Moving on, because I can see I'm running out of time, so I'm going to skip over my next slides. Just to say that I think there will be a paucity of clinical evidence for rare diseases, and so the other forms of evidence are important. And something that's very close to my heart is patient-based evidence, because patients can share their experiences, their perspectives, their attitudes, preferences, values, and expectations, not just about a new medicine, but about their current care, about current pathways. And all of this can be captured through participation, which is increasing, but through good research as well, through quantitative research and through qualitative research. And often qualitative research is labeled as being anecdotal. This does a disservice to this really good science. Cochrane is about to produce a whole new handbook on qualitative evidence synthesis, which clearly describes how it can be critically assessed. So we need to be incorporating this kind of evidence into our decision making. And finally, I'm going to end on the next slide on economic modelling, because that's used by many countries. And just to explain, so cost utility analyses rely on long-term modelling of the costs and consequences of a new medicine and its comparator. And this requires definition of disease states and the way in which patients progress through those disease states. And then for each disease state, there is a quality of life utility associated with it. Now, this is a real challenge for rare diseases because often we don't know the disease states, and we have very small numbers of patients progressing through in these models. There has been research to develop these utilities in rare diseases, and a range of approaches exists. But if we do another click on this slide, you'll see that there's a real challenge for me, because quite often we see economic models coming forward and clinicians and patient experts say all the disease states are wrong. They just don't make any sense at all, and they're not how experts see the disease progression. And yet those models are still used for decision makers, because the decision makers say, well, any model is better than none. I would query that. I would also say that extrapolating for a heterogeneous rare disease is also incredibly challenging. Moving on to my last slide. I would just like to say that some of the work I've drawn today is from the Rare Disease Interest Group, for which we have 4 projects outlined here. And I'm delighted that Alicia chairs my project group 3 on new paradigms of evidence generation and realistic interpretation of evidence. And I look forward to further discussion on these points in the discussion section. Thank you.
Thank you, Karen. I think you have made a very, very clear explanation on how HTA approaches value assessment and also challenging the uncertainty word, how it is used as well, and misuse, and where traditional frameworks can struggle in rare disease. At this point, I was planned to talk to Florian and give him the word that is the most directly affected patient perspective. It's so important. But given that Francois is here, and I don't want to miss his experience, he's, let me say, a kind of unique animal, because he's a physician. Let me, sorry for being so direct, and is a physician, has been a regulator, has been an HTA, and now it's a very senior consultant helping people to better cooperate. So, Francois, I'm challenging you, but I think we missed your first presentation. If you can adapt what we have been hearing, we have been hearing a regulator, we have been hearing an expert in HTA, but critical with HTA and rare diseases. And now you, before Florian will leave with his presentation. Francois, the floor is yours.
Yes, thank you, Alicia. If I may have the first slide. Sorry to have, for this problem, blame either Microsoft or the company the computer I have, who left me without the possibility to do anything yet. And I felt a bit like a pianist called Maria Jo Pires, and there are videos on the net where she's intend to play a Mozart concerto, and then the chef starts the music, and she realized that she's prepared for another concerto. And it's such a drama for her. She looks absolutely devastated. So I looked devastated for a while, but now I'm connected through 2 computers. So if there is a problem, tell me, I will switch to the other one. Now I have to introduce the presentation you already had. So I'm here to say the general things that not of a big interest, but introduce you and give you appetite for listening to the others. So now I don't know how I will do, so I will be a bit quicker to leave room for the discussions. So if we look, that will be my first message, having been involved in various steps of the drug production or development or evaluation, is that if you looked at the adoption of a new medicine, the agenda, we have a lot of actors, of stakeholders. We need an inventor. We need to have to fuel the development with investors. And now we have developers, which are pharma companies, that prepare the data to request for the next step to present to the regulators the data, and the regulators ensure that we have a safety and efficacy that allow the drug to be on the market. And then we have health technology assessment, the HTA, that assesses clinical or economic value. And then we have the decision makers. We call them payers, but it's not a good term. They are decision makers in terms of pricing and reimbursement. And this final decision is capital, because it's these decisions that allows patient access to the new evaluation, to the new drug after the evaluation. So these, and of course, these are institutions, but we have stakeholders that are of utmost importance, patients, which must be at the center of all stages. And Karen just mentioned how we should make some progresses in the fact that the information we get and the input we get from the patients, clinicians that of course are the interface between the science and the patient themselves, and also citizens, because they are the real payers and they should have a voice. And sometimes you can hear somebody or clinician or patient, a clinician saying, okay, it's good to pay 2 million euros a year for a drug for a rare disease, but why why I can't have 2,000 euros that I need to take care of a young adolescent who just committed suicide? And of course, they are good questions. So we need to take all these aspects into consideration at the political level, the policy-making level, and at the political level. Next slide, please. So we will focus on these two aspects, regulators and HTA, because we do not have payers to discuss with, as it's a bit more difficult to discuss with payers than to discuss with regulators and HTA body. But we should remain that it is a chain, and the chain is not stronger than its weakest link. So if finally the payers are reluctant to take on board this new drug because they think there is maybe either a lack of knowledge or uncertainty or whatsoever, they do not like uncertainty. So that, of course, will prevent the patients from accessing the drug. Shall we go to the next slide? So then there is the fact also that we have these two evaluations, regulatory and HTA, but for the marketing authorization, it is at the European level with the EMA, and for the HTA and the decision making, it's done at the national level, and there are various levels, various organizations for HTA in each member state in the European Union, or in each country. It could be agencies, committees, the ministry, whatever. So we have various systems. The criteria may be a bit different. They need to take into account also local factors Karen mentioned, and there may be sometimes regional difference for availability of new drugs. Next one. So the marketing authorization, I will not develop, just say that the company submits a dossier, EMA evaluate benefit risk and concludes in favor of marketing authorization if benefits risk ratio is positive, but there is it's not in the mandate of the EMA to compare the new drug with existing ones. So it may be better or not than existing ones. Even it can be not as good as previously existing drugs that are already in the market, provided that it brings a useful alternative, for instance, for patients who do not tolerate the more efficacious drug that is on the market, or for which they have not responded to this one. So no comparison in the marketing authorization. Next one. So on the contrary, the HTA process is comparative. So could be on the basis of economic evaluation or clinical evaluation. So in some countries, it's based on the clinical evaluation, like in France, Germany. So the conclusion should be whether this drug brings added clinical benefit over existing treatment. And these conclusions as consequences of on the decision for reimbursement and pricing. And this is the final part of the process. This is the appraisal phase where the committee look at the relevance and the significance of the data provided and make a judgment, because at the end of a scientific process, there is a judgment whether it is significant or relevant, and they conclude on the eligibility to reimbursement and clinical added value. It's good. And this can be based in many countries in economic evaluation, like Karen showed it. But before that, we have an assessment phase where you collect the data that have been submitted by the company, you can complement by the literature review, and then you analyze and indicate whether what are the data provided and whether there are some methodological problems or not in the clinical trials or in the clinical information submitted. So we have two phases, assessment and appraisal. And important to know because now HTA is still national, but a cooperation has started first voluntarily during more than 10 years with the UNETA project, and now since the implementation of the HTA regulation, now it is mandatory currently for a part of drugs and progressively for all drugs, and the next ones to come will be for the orphan drugs, to produce a joint clinical assessment. So not anymore nationally produced assessment report, but joint clinical assessment reports. Next slide. So what are the challenges? I will not insist because it has been said and probably everybody in the audience knows that, but this small limited sample size, heterogeneity. I will select 2 points. First, the point that it is difficult or not feasible to conclude randomized controlled trials, and also the problem with appropriate comparators, and because sometimes these comparators are, for instance, off-label use of some drugs, and that raises problem. Next slide, please. So high uncertainty, okay, we say economic evaluation challenges. Finally, what we have a tension between not this, maybe according to the historical ways to assess evidence, it may consider to be a low evidence data, but there is an unmet medical need in very severe disease, because rare diseases are severe diseases. So there is a need to have a balance between the rigor, methodological rigor, and ethical public health consideration, and consider the fact that what we have to produce is something that will be helpful to the patient. We do not treat a disease as a physician, we treat a patient. And that's for that reason, really the patient should be really at the center of everything and should be taken into consideration during all the assessment process. Next slide. So just some thoughts about the off-label use of a comparator. That's a problem because off-label use of a comparator, you cannot do a randomized control trial using as a comparator an off-label use. It's not ethical, you cannot do that. So comparison is necessary because it may be its parts, and it's mandatory in many countries to take into consideration off-label use. In the past, it was not the case, but now almost everywhere, it has to be compared, to be taken as a comparator, the off-label use of a drug. And joint clinical assessments will make this more and more necessary, because the joint clinical assessment is on the comparative assessment, on the efficacy. So you need to put comparison in the data you submit to the Europe for the production of a joint clinical assessments. So how do HTA bodies handle this situation? Next slide. So just an example, in one country, that was a poster that were presented in I think it was ISPOR Europe 2024, 2 examples in Portugal. One study has concluded that the use of off-label comparison was not detrimental for the final appraisal of the drugs, and the other had the opposite conclusion, saying that if the comparators in off-label use of a drug, this decreases the chance of a positive outcome of the HTA report. So things are complicated. Even this small thing may be quite complicated to handle. Next slide. Now, randomization, it's very important. Randomization is the only way to take into consideration known biases, but also unknown biases. I'm old enough to have known a time where drugs were put on the market without necessarily good quality data, not necessarily randomized controlled trials, but a tenured professor saying this drug is good, I've given it to my patients. So this should not be, this of course was not good, and has been replaced by the fact that all drugs should show randomized controlled trials. So now we see that it's not the case. Single-arm trials are produced. And so what is now currently the situation when you want to submit non-randomized studies to HTA bodies? So this is an example from a publication from my former colleagues from HCS. Next slide. So again, it's RCT remain the gold standard for HTA bodies for estimating unbiased treatment effects. And so what they propose is alternatives, alternatives to the fact to produce non-randomized clinical trials is to have adaptive designs, platform trials, all the things you see listed. So it's better to have randomization in a sort of simplified way, if can be done, that's to produce non-randomized controlled trials. Next slide.
Francois, in order to make it possible for Florian to intervene as well, could you...
I conclude. Okay, I wrap up.
Thank you.
So the fact that the circumstances in which non-randomized controlled trials are very limited, and that the criteria for that are not made public. So it has to be assessed by the HTA bodies on a case-by-case basis, and you don't know when you develop whether you are good or not. You will know that only at the end. So that's something that is a bit difficult, and that should, I think, change. Next slide. Okay, so that's the European cooperation on HTA, the regulation. So we have, to the right, the joint clinical assessments. I've explained to you what it is about. There is no value judgments in there. These remain at a national level. So we cannot expect to have a common appraisal for a new drug because the appraisal will remain at a national level. And then we have the possibility to discuss the development plan in the opportunity early before deciding the clinical trial design with a joint scientific consultation. Next slide, please. I think it's reaching the end. Okay, I will not. This is just an extract from the guidance on validity of clinical studies from the HTA regulation, but I have nothing particular to say for that. Next slide. And I think so. Just to we need cooperation, such as happened in the congresses and such as this project and other projects that deal with rare disease or deal with field effectiveness, what sort of things, because we need to take into consideration all type of information. And the rare disease information group has been presented by Karen. Next slide, I think at the end. Okay, I will not mention, it's just one of the designs you can adopt. And the question would be, has this design be used for the new drug presented to the EMA? Does anybody knows this type of trial, and what is the potential development of such a methodology? Next slide. Thank you for your attention. Sorry to have been a bit long.
No, thank you. Thank you so much, Francois. I really appreciate the flexibility of everyone here in order to adapt to the technical issue, especially Florian being patient on this. But I think it's essential. It's essential to have the perspective of the most directly affected. And I think, Florian, please share your patient advocacy perspective in the way you prefer. I think there is a lot of meat here, even for another webinar, but please try to summarize, because also we would like to hear what are the Q&A people, what is people saying. But first, yours. The floor is yours.
Thank you very much for the presentation or introduction. Yeah, I'm going to start right off with some food for thought. First topic would be RCTs and rare, because not every condition is rare doesn't mean the same thing everywhere. Karen pointed that out earlier. And cystic fibrosis is actually a pretty good example for that, because on the one side, you have one of the more common mutations, the 508del, which consists 65% of every person with cystic fibrosis. But there is a whole bunch of other mutations, a couple thousand of them, which make up the rest. So you have in one condition the possibility of having a rare and ultra-rare at the same side. And for some, it's totally possible to have a randomized control trial, like for the common mutation, definitely. For the rare ones, with let's say 20 people in Europe, not so much, right? So in some of these conditions, RCTs are definitely possible. So I would like to stand against that statement that RCTs are not possible in rare diseases. Next thing, or next bullet, please. The next aspect would be uncertainty in rare. Because in the end, I'm coming for a growth disorder sector, and expecting in an HTA assessment final height data would mean for a drug that is during puberty or during the growth stage applied, or is applied during the growth stage, we would need trials that endure for 15 to 20 years, which is longer than the market exclusivity or the patent whatsoever, which just means that is totally unrealistic. So we have to deal with that. Next slide, please. The impact on the rare community. Francois pointed out a couple of things. The first one would be the European Cooperation HTA, EU HTA. The expectation management due to the fragmentation in Europe is tremendous on the patient side. And it was one of the key aspects to implement that process in the first place, to get greater access. But is it really an assessment? And Francois actually pointed it out at the first bullet. Alex, please just put the bullets on a little bit. Thanks. It does not conclude anything. It's going to be a descriptive description of the results, but not the verdict in the end. Something that I would expect from an assessment. Rare is to be considered, but it's open to discussion. How? And Francois also pointed that one out. It is by design a comparative discussion. So single-arm trials will result in indirect comparisons, and given the methodology guidelines, we will see a lot of discussion regarding those comparisons. Long story short, as the results and reimbursement decisions are going to remain on the national level, it is somewhat questionable that this process is going to result in a greater access for patients within the European context. Nevertheless, it is a good start because it's better to have an assessment for especially for the smaller countries which do not have an own HTA body. At the same time, there's the pharmaceutical package, which has not been discussed yet in this talk. It is currently in discussion, so it's difficult to say anything in that regard, but there are some couple interesting aspects in the proposal. Next slide, please. So what do we need from a patient perspective? First of all, we need patient participation in trial and endpoint design, and from the beginning till the end, because it is crucial that the people who give input at the beginning in the endpoint design know what's going to happen to exactly that endpoint in an HTA assessment, which can be something totally different. And it's crucial to know that, especially when it comes to the interpretation of endpoints. We've had discussions in the Federal Joint Committee around a lot of things, especially endpoints in rare disease communities, obviously. And sometimes it's hard to say what a specific change actually means for a patient. And these things could be easier if there is an agreement on what these actually mean, and patients could deliver a valuable perspective on that. What we also need are comparisons. And I'm very keen on that. I don't really like single-arm trials, not because I don't like them, but we need a comparison. And it could be an external comparison, solidly done. I have nothing against those. But as a patient, I want to know what the specific drug does and how it is compared to whatever there is except that. So, and obviously solid trials, what I mean by solid trial, when we're talking about a chronic condition, a 4-week trial does not really fit my standards, to be honest. Same thing, active controls as soon as possible, because having placebo-controlled trials for a longer period of time do not really make sense from a patient perspective, because we really want to know the same thing as an HTA assessment. Is it better or worse than what is out there? And that may be an active control. What we also need is an expectation management on all sides. That means for the pharmaceutical companies being open, what is possible, what is in line, what is not in line. Doctors being open about specific trials, what advantages there are, what disadvantages there are, so people can make an informed decision. Very important, end of trial is not end of research. That has been said a couple times, but it is crucial for us, especially when it comes to uncertainty levels that I've discussed earlier in terms of, let's say, end of growth data or final height data. And that results, at least for me personally, in a data collection and registries. And I would favor actually for them to be mandatory. Next slide, please. I do have a couple wishes for the future because I would really like to be in discussion with all of you. I would love to have a shared understanding of trials from the beginning to the end. That's one of the reasons this project is interesting, because it brings together all of the stakeholders, not only industry and academia, but also some of the HTA bodies and the regulators. So it's really good that ICWIC is here, not in this project alone, but here personally I've seen one of the representatives. And another thing would be an answer to the question, what level of uncertainty are we going to accept for specific conditions? And that might differ actually from rarer to ultra rare conditions or within conditions itself. So thank you very much for the attention, and I'm looking forward to the discussion.
Amazing, Florian. I think has been the perfect end of this conversation, which at least it shows very clear that advancing in evidence generation and assessment in rare is not only about new methods or frameworks, or it's about genuine shift in mindset across developers, regulators, HTA, and trying to understand how to align better. I don't know if there is any. I have a lot of questions, but I really would like Alex to let us know what people is concerned about. What are the questions? Or is anything that you want to clarify, any of you of the speakers after hearing Florian? Or I love how Karen was challenging the uncertainty concept in rare. I love, and then Florian, but let's see if you have any urgent clarification or questions before Alex give us idea what are the Q&A insights. Any of you? No?
So we have one question at the moment.
Okay.
Alicia, which asks, are rare cancers included in RealiseD?
Say again?
Are rare cancers included in the RealiseD project?
Good questions. Very good questions. In fact, this is not discard, but we really wanted to focus on not non-cancer ultra rare. But you know, we are still in the first year of the project, and this is time for shaping. I think I'm the only qualified to answer in this moment in this, because I'm the one. But I'm for sure that if there is any other RealiseD colleague in the audience, please feel free to think. In my opinion, when we were revisiting pathways, we were not focused on cancer rare for the moment. This is my perception. So if there is, Karen, tell us.
Yes, I was just going to raise this. It's an interesting question because it's one that's been raised to the HTA Rare Disease Interest Group. And I think our thinking from the beginning was that, no, we're actually focusing more on the kind of genetic rare diseases rather than cancers. And a key reason for that is there are actually fewer uncertainties with rare cancers and fewer unknowns. So many rare cancers follow standard cancer pathways. So many countries have approaches that apply to all cancers. They can be more difficult to diagnose, so they have that in similarity, but often the outcomes are tumors or hematological outcomes. And so there are a number of more known things about them. But thinking about our own work in the HI Rare Disease Interest Group, that characterization kind of compendium could be used. So we could say, yes, this is a rare condition, but actually we know a lot of these things. We know the care pathway, we know the outcomes. So there are fewer things that we don't know. So maybe you need less leniency compared to, for example, a childhood genetic rare disorder. So it's a tricky area, but I think that in general, there is more known about outcomes and there's better data collected, etc. So we focus more on the really difficult areas.
Thank you, Karen, for the clarification. And also, I think the focus on ultra rare, it's a model that could be translated later. But I see Solange also with a hand raised, please. Welcome and go ahead. If you have any questions or comments.
Oh, thank you. And thanks to all, what we have heard, so far, fantastic experts. So it's definitely, quite amazing for RealiseD. And I have to say that indeed, rare cancer can be part of RealiseD as well, knowing that the focus of this project is all about rare, ultra-rare diseases, but it is also mentioned small population, such as, for example, children with cancer. So then, the answer to the question which was raised is yes, for sure. Thank you.
Thank you, Solange. Sean, I see Sean Tunis also wanted to, I think it's a comment, not a question. So Sean, any question, comment?
Yeah. You can hear me okay? Yeah.
Yeah.
Quick comment. I think it was to something Florian had mentioned, and then a question also related to Florian's comments. All the comments were great, by the way, so thank you. The comment is about, the feasibility of randomized trials in rare disease, and I think, probably there's agreement that it depends on how rare the disease is. So that, and I thought that the framework that Karen, you showed from the paper on which Hans-Georg is one of the authors was a nice framework for sort of determining the feasibility of randomized trials in rare disease, looking at a number of features. So I thought that was quite helpful. The question was, I think it was on the last bullet of the wish list about, defining an acceptable level of uncertainty. And I would love to hear comments from the panel on how does one even begin to think about that question? Because it's a puzzle for me. So I'd just love to hear any thoughts on it. Thanks.
Sean, anyone want to answer? Yeah, I see first Francois and second Karen, and third Florian. So good. Go ahead, Francois.
Very quickly, I think the level of uncertainty you're going to accept, because it's true that lack of knowledge may be different from uncertainty, but for the decision maker, it's the same. It's an information they don't have in their hands, so it's a problem for them to make the decisions. And this question to be answered needs to know how can we decrease the level of uncertainty? How can we increase the level of knowledge? And do we believe in these methodologies? And are we able to decrease that over time and then to assess a drug not only once, but over time and refining the way they are used or reimbursed or whatever?
I think it's fascinating because I think it's more inherent in the deliberation than it can be quantified. So I think if you observe the way decisions are made for different rare diseases, it is obvious that conditions that affect children, particularly infants, that are extremely life-limiting and often very disabling, in a sense, greater uncertainty is permitted in those rather than a chronic disease which is maybe lifelong and actually isn't very limiting, but is more about quality of life. And I think that's what we're trying to do, is to try and characterize some of those things so we can be more explicit about those. And it's that this question is, what is the alternative? It's a bit like in ethical considerations around rule of rescue. So there's often a rule of rescue that comes in here, which is about, you know, if a car accident turns up, somebody who's been involved in a car accident turns up at the emergency room, they will get immediate cover because that's the rule of rescue. And I think that we feel that rule of rescue with certain rare diseases, but maybe not with all. And I have to, we're probably not allowed to talk about this, but sometimes even when you think the rule of rescue might apply, the price is just too high for the health system given limited resources, and that's an unfortunate ethical situation that we're in. And I think we need to be more explicit about these ethical trade-offs that we have to consider from a societal perspective.
Thank you, Karen. Florian?
I totally agree with Karen's comments, by the way, that there is a change within the, it depends a lot on the condition. Some of the late assessments that could be done in some systems have a certain disadvantage. And especially in those conditions in which a lot is happening, the question might just change over time. So if you have the data 7 years later, the question is totally different and the answers might be different. So that might be something that needs to be considered from my point. Later said, because a lot of times we're talking about early assessments and that automatically means a higher degree of uncertainty.
Thank you, Florian. We have Philip. Can you identify yourself for the sake of the rest of the people to know who you are?
Yes, thank you. I'm Philip. I'm working at IQWIC, HTA agency in Germany. And I also wanted to give my opinion about the uncertainty. And I agree with a lot of points which were made already. But I think it's always important to see that if you don't have the data, the uncertainty is high. And this doesn't differ between non-rare or rare diseases. So if you don't have the data, you cannot come to a data-based conclusion on efficacy or effectiveness of a new drug. And I think at some point, you have to make clear that you lack this data and it's difficult to come to a decision based on this limited data. And as I said, this is not different for rare diseases or non-rare diseases, but at some point you come to the difficult decision, and then it might be more an ethical or a societal question you have to answer, which is how much uncertainty you are willing to accept in a certain situation where evidence is limited. And first, you have to appraise the data you have. And if the data cannot give you the opportunity to come to a clear conclusion, you come to different questions, which are also part of HTA, as Karen mentioned, the ethical ones, the societal ones, the monetary ones, of course. But I think the data stays the data, and what you can do with data is not necessarily different if the indication is rare or not rare.
Thanks, Philip. Any of you want to react to this? Okay. I will leave Sean to say something because it's the way how you consider data. But go ahead. I don't want to go in a dialogue.
I promise
Sean.
it's my last question. So the question is, oh, and by the way, I'm Sean Tunis. I'm kind of in the HTA field based in the US. I've worked with Alicia and Karen and Francois for many years. The question is, I think, Alicia, you used the term maybe in your introduction, best possible evidence. And I'm wondering to what extent the degree that this question of uncertainty depends in part on how well the product developer, the researcher, did compared to what's best possible. In other words, is the sort of if you could have done a better, if it would have been possible to generate higher quality evidence with less uncertainty, does that sort of mean, factor into how acceptable the uncertainty is, or if you've really done the best possible given the limitations in terms of the nature of the disease and the therapy? And I just be curious if that's a factor in the appraisal, and acceptability of uncertainty.
Thank you, Sean. I think there are reactions from Karen and from Andrew. Karen?
Yes, thanks, Sean. Actually, it was fascinating for me when it's in the definition. It's in the definition of HTA, this issue about best available evidence. But what I've noticed over the years is certainly in the HTA body in Scotland, we only have like a paragraph now about the technology. And describing the technology and the condition used to be important. And if we spent more time kind of doing what the regulators do, the regulators go into all the background and the mechanism of action, etc. We don't do that so much in HTA. We kind of are following on from the regulators and looking for other evidence. But I think we need to take a step back and clarify, in a sense, the scientific basis of the disease and the mechanism of action of the treatment. And then when we know that, I think we can start judging about what is possible and therefore what is best available. So I would love to see that step also being more prominent in HTA of rare diseases.
Thank you, Karen. Andrew, you want to add something?
Yeah, thanks. There's a regulatory, something of a cliche that you can only assess what's in front of you. And obviously, if a company has come in and sought scientific advice and has been told this could be acceptable, even if it's not the best available package, then it's difficult for decision makers to go against their own advice further down the line. I think another thing that does need to be taken into account, and I have seen from an ultra rare perspective, is what is the reasonable chances of getting other evidence on top of this. And there are some ultra rare diseases where the development program has already used 40% of patients globally for a single arm trial. There isn't really another data source or set that's realistically going to provide any additional information in a reasonable time frame on top of what's already been done so far. So yes, I think it's a combination of what's been done, but also what can be done as well on top of that, and potentially as well within the context of a post-licensing commitment as well, and not necessarily something that there is uncertainty and that could be addressed on the market rather than waiting longer, and that would depend on the nature of the uncertainty at hand. Thanks.
Thank you, Andrew. I think it's Florian wanted to react as well, and then Francois, and then I will make the last question for you.
Just a quick comment. Sometimes it feels like it could have been done more by companies when the trial design started. That some of the cohorts, that external cohorts or historic cohorts, could have been planned earlier and could have included better and more robust data, which would make an assessment later on a lot easier. So if you are a company, please consider doing that better and earlier. That would help sometimes a lot. Thanks.
Thank you, Florian. And Francois? You are on mute.
The question of the price that Karen mentioned briefly, of course, is important. The higher the price, the higher the reluctance of the payers will be to accept a new drug with some uncertainty. That's just a remark. Another remark is a question I have. I think I've seen in one of your presentation, Alicia, that you mentioned also the mechanistic reasoning to be one of the tools to be used, and I was interested in that, and that's interesting to see that there are publications. In the past it was a nightmare because a drug could be authorized just because there was a mechanistic reasoning saying that it may work, but now mechanistic approach can complement, and there are studies linking mechanistic conclusions on clinical outcomes, and that's been done for Glivec years ago, and that's quite interesting. And the last point I wanted to make, I think I forgot it for the visuals again.
No, I think I really appreciate, because in the end, the title of the webinar calls for a mindset shift. It looks like there is an agreement in the air that we need everyone, developers, regulators, we need to do better what we are doing. We need early alignment, but we still, in this conversation, it's clear that still how uncertainty is perceived, understood, make the most important element for decision making. So from your perspective, what would be the single most important shift that still needs to be happen between all the actors here, developers, regulators, and HTA bodies? This is, I would like you to think quickly, what is the most important shift that still needs? And then, given that we are in this RealiseD project, we have 4 minutes to leave. What will be your recommendation for this project? Because the project of its RealiseD project aims to contribute to this shift in fostering dialogue, alignment, and blah, blah. So please, let us know. I can start by Francois, then Karen, then Andrew, and Florian. Yeah. And Alex, I don't think it's any other question. No? No. Okay. We are doing a nice conversation here. Okay. So let me, single most important shift still need to happen.
Yeah. And I think we need to consider the HTA regulation very closely, not because it will change everything, as Florian reminded us, it's just an assessment, there's no conclusion, so everything will remain specific, but however, there will be a joint assessment, so we can hope that there will be more common approaches on the way to analyze the evidence available for orphan drugs, and we should take advantage of more dialogue and to have some step ahead on that. My other wish is that, because I'm monomaniac, of the involvement of payers, because, that's the final link and we need them on board, and that's not that easy.
Thank you. Andrew?
I would say clarity and consistency from all decision makers with their expectations for developers and for patients, so that people developing drugs know what needs to be done and patients are aware of what has been done and why the evidence base has been deemed sufficient for licensure and reimbursement.
I think I did realize it were package 6, absolutely.
Well, I think Alicia said, with respect to RealiseD specifically, a lot of the kind of technical work packages, looking at some of the more methodological aspects, really make it clear what are the assumptions, what are the that are being made when modern methods are being used. How are they being implemented in practice? What is being let go of compared to the usual clinical trial design that more appropriate for rare disease designs are offering instead?
Thank you, Andrew. Karen?
Yeah, specifically for RealiseD, I think it's very impressive research being undertaken to support clinical research. And I would ask that doesn't just have a clinical focus, but thinks about the downstream decision makers of the regulators, the HTA, and the payers, which I know you're trying to do, but does that within the design context. So, for example, recognizing that clinical research and clinical studies will be part of an evidence package. So how does that fit with complementary real-world evidence? How can clinical studies be designed to have sub-studies that look at patient-based evidence, for example? to do simple exit interviews to understand the differences it's making to patients. And the economists would say, please include EQ5D in every clinical research study. So just making sure that if we're doing small trials, we're trying to include the outcomes that are needed for the downstream decision makers, not just for regulators.
Thank you. Florian.
Well, actually, it's projects like this that I have high hopes in, because these projects bring stakeholders together, and especially if it's all stakeholders and not only parts of them, we have the possibility to get people together and get things moving. And that's, I have hope in the end.
Well, thank you very much to all of you, the speakers, for such a rich and thoughtful discussion and conversation, and to the audience for being patient with our technical things. And with that, I would like again, thank you for joining us today, and formally close the webinar on time, I think. Thank you so much.